Welcome to the lecture on cell transport. Before we get into transport, there are lots of different ways that transport can be described, and we're going to break those down using these vocabulary terms. Materials move across the membrane in a variety of ways, and each of those ways can be put into one of two groups, passive or active. If the type of movement requires no energy, then it is passive. These are based completely on a concentration gradient. Active transport is when items move against a concentration gradient, so energy is going to be required. We'll learn about what that concentration gradient means in just a moment. Facilitated diffusion is a type of passive transport. No energy is required, but it does use helpers like proteins to move things in or out of a cell. That's how glucose gets from your blood into the cells of your body. It's too big to move through the membrane, but is passively moving in due to a high concentration on the outside and a lower concentration on the inside. The two most common types of passive movement are diffusion and osmosis. The difference between those two is that diffusion is the movement of solutes and osmosis is the movement of water. We're now going to break down what a solution is. That's when there are substances called solutes dissolved in a solvent, which is usually water. Think about this. If solutions are solutes dissolved in a solvent to form a liquid, a good example is Kool-Aid. What in this scenario is the solute? What is the solvent? And what is the ending solution? The solute would be the Kool-Aid powder. The solvent is the water that is dissolving the Kool-Aid. And then the end solution is the Kool-Aid itself that you would drink. If you're having trouble answering these questions, please make sure that you contact your instructor so you can get clarification. Diffusion is the movement of a substance down a concentration gradient. And a concentration gradient is when there is an area of high concentration next to an area of low concentration. A gradient is any time that there's a difference between two areas. We can also use the gradient to describe a hill, where there's one area that is high and one area that's low, and there's a gradient between them. Concentration gradient is the same thing. It's just speaking about concentrations rather than elevation. So no solute likes to be crowded, therefore it's going to move from an area that's very saturated or has a high concentration to one that is not saturated or has a low concentration. The process of spreading out is called diffusion. So I'm sure that you have been in a classroom where someone's sprayed perfume. And you might not smell it at first because it was over in the corner, but the perfume will spread out throughout the classroom. And pretty soon you'll be able to smell that perfume throughout the whole room, even though they only sprayed it over in one corner. That is diffusion. The perfume is moving from a high concentration, where it was sprayed, to a low concentration, which is the rest of the classroom. This doesn't take any energy, it's just the natural way in which molecules move. Osmosis is the passive movement of water. I have a picture of a slide on here to demonstrate how things move from high to low concentration. This slide represents a gradient, where one area has a high concentration, one area has a low concentration. If you go down this slide, that doesn't take any energy on your part. You're just moving from high to low. Same thing with these molecules. As they're spreading out, there's no energy required in order to do that. So let's practice. You might have some questions on assignments and tests as to which way a solute would move. Here we have a picture that shows a semi-permeable membrane in the middle. The big blue circles are solute, and the little red circles are water. If I were to ask you which way the solute would move, that would move from an area of high concentration to an area of low concentration. So they're moving in this direction. Make sure not to use words like up or down, because there's not that possibility for that movement here. Look at this next picture down here. Which direction do you think that the solute would move in this case? Make sure to always pay attention to your key. Here, the solute is the smaller white circles, whereas the water is the big blue ones. You should always pay attention to that because it's the opposite of how the key is up here. In this case, there are fewer white circles on the inside than there are on the outside. So we would move from an area of high concentration to an area of low concentration. So into this circle, or pouch, or cell. Again, make sure not to use words like up or down, because that could represent any direction here. I could move up into the circle, or up out of the circle, or down into the circle, or down out of the circle. So there's no up and down are not appropriate words to describe movement here. You would want to say into or out of the cell. So how do you think these processes could be sped up, or how do you think we could slow them down? Take a minute and think about what you already know about how molecules move and what could possibly make them move faster or slower. Some ways you should remember are that we can speed up this process by increasing the temperature, increasing the pressure, or increasing the concentration gradient, the amounts of solutes. Active transport is when solutes move from an area of low concentration to an area of high concentration. So therefore, it requires energy. That's like trying to run up the slide as opposed to going down the slide, or trying to put the smell back into a can. It's going to take energy in order to do that. So active transport moves from an area of low concentration to an area of high concentration. Now that we understand the passive and active transports, we are going to use some terms to describe types of solutions. So hypo means less and hyper means more. Tonic means solution and osmotic means water. Iso is equal. So if there is more solution or a higher concentration of solute, we would use the word hyper for the more and tonic for the solute. So hypertonic describes a solution that has more solute. Less solution or less concentration of solute, we would use hypo for the less and tonic for solution. So one that has less solute would be hypotonic. Equal amounts of solute, iso is equal, solution is tonic, so isotonic is when two are equal. Remember, these terms are used to describe comparative areas. You can't just say that this Kool-Aid is hypertonic. It has to be hypertonic compared to something else, like water. Kool-Aid has more solution than water, so therefore it will be hypertonic compared to the water. But we can't say that use any one of these terms without using it as a comparative term. So if there's more solute, then what does that say about the amount of water? If you have a container of really, really strong Kool-Aid that's very hypertonic compared to other types of Kool-Aid, you can kind of think of it as that there's not as much room for the water in there because the Kool-Aid is taking up all of that space. So if there's more solute, that means that there would be less water. So we can say a hypertonic solution is also hypoosmotic. A solution that has less solute would be hypotonic, but that leaves more room for water. So that would be hyperosmotic. The amount of water and solute are opposite of one another. So if there's more solute, then there will be less water. And if there's less solute, that means more water. So this affects the way that water moves in any given scenario. Osmosis is when water moves from high water concentration to low water concentration. So if we look back at these same diagrams, we can see that there's more water on this side and less water over here. So water would move from high water concentration to low water concentration, like this. Notice it's the opposite of the way that the solute would move. Osmosis and diffusion will always go in opposite directions. Which way would the water move in this other scenario down here? In this scenario, it's not as obvious as to which side of the membrane contains more or less water. They look to be the same. But remember, we said that our diffusion was movement of solute from high to low. Osmosis is always the opposite. So this means that water would move out of the cell in this direction. Remember not to use terms like up or down because up is relative. Could go up in or up out. Use into or out of. In this case, out of. So this is very important for our cells. Because if you put a cell into a solution that's not isotonic, there's going to be consequences in what happens to that cell. That's one reason why when you have an IV, they put saline solution into your blood, not just water. If they put just water, that creates a hypotonic solution on the outside of your cell. If there's a lot of water on the outside and a low concentration of water inside because your cells are filled up with solutes, water is going to move through the membrane into your cells. This can cause your cells to expand or burst, and that would be a bad situation. The same problem could be said for a hypertonic solution. If they put solution into your blood that is too salty or has too many solutes, then we have lots and lots of solutes on the outside and not as much on the inside of your cells. So water is going to leave your cells in an attempt to stabilize the internal and external environment, or intracellular and extracellular. That's going to cause the cells to shrivel and even die. The best scenario for our cells is an isotonic solution, where the concentration on the outside is the same as the concentration on the inside, so water moves in and out equally. Water movement doesn't just stop altogether, it's just that equal amounts enter and exit the cell, so that its size stays constant. Both hypertonic solutions in your blood and hypotonic solutions in your blood would be bad for your cells. You should be able to determine which is which. So make sure that you understand the hypertonic and hypotonic solutions. If you're not able to do these, make sure that you contact your instructor for clarification. So if a cell loses too much water, it will shrink and it can die. This is called cryonate or plasmolyze. If a cell gains too much water, then it can burst. It's called cytolysis or just lysis for short. Both of these things are bad for cells. Cells want to maintain homeostasis. Remember that from the last unit? Where homeostasis means a stable internal environment or a biological balance. We want the solution to have the same concentration outside as inside, and so water can move in and out freely. The last types of movement are movement by vesicle. Ask yourself this question: what is a vesicle? A vesicle is a pouch, a membrane-bound pouch, and it can hold a number of things. Lysosomes are vesicles that hold digestive enzymes. But vesicles that we're talking about here are basically just going to contain pouches of whatever's needed. Vesicles can move things two ways: into the cell or out of the cell. Endocytosis means that it's bringing things into the cell. Exocytosis is moving things out. It can also move two different types of materials, liquids and solids. Pinocytosis is moving liquids, either in or out. Phagocytosis is moving solids, either in or out. So these are not exclusive. You can combine them to describe different types of movement, like endopinocytosis would be moving liquids into the cell, or exophagocytosis would be removing solids, pushing them out of the cell. That's all for the transport lecture today. Please scroll down and complete the assignments that are for the rest of this week, and contact your instructor if you have any questions.